The possible neuroprotective effect of pomegranate extract with L-DOPA in rotenone-induced Parkinsonism in rats, represented by Jilan Ala Kamil, Lecturer of Pharmacology and Toxicology, Faculty of Pharmacy, Al-Azhar University, Cairo, Egypt. Parkinson's disease is the second most common neurodegenerative disorder after Alzheimer's disease. It is characterized primarily by motor symptoms such as tremor, rigidity, and bradykinesia. It is associated with degeneration in nigrostriatal dopaminergic system. The exact etiology of neuronal degeneration in the Parkinson's disease is still unknown, but it may involve several cascades of events, including oxidative stress, inflammation, pro-apoptotic mechanisms, and mitochondrial dysfunction. L-DOPA treatment has been considered to be the standard therapy for alleviating motor symptoms in Parkinson's disease. However, during progressive treatment, L-DOPA becomes gradually less effective and generates motor complications such as dyskinesia and dystonia. Therefore, the current practice is to provide neuroprotective drug with lower doses of L-DOPA. Pomegranates are very rich in different polyphenols with antioxidant and anti-inflammatory activity, which are likely responsible for the beneficial properties of the fruit. Pomegranate extract also has shown beneficial effects against neurodegenerative disorders such as Parkinson's disease. Aim of the work: the present study aims to study the importance of adding pomegranate extract as a potential agent to lower dose L-DOPA in rotenone-induced Parkinson's disease model. Study the possible underlying neuroprotective mechanisms. Design of the work: 40 adult male albino rats were divided into 5 groups. Group 1 is the control group. Group 2 is the rotenone-intoxicated rat for induction of Parkinsonism. Group 3, rotenone-intoxicated rat treated with L-DOPA 25 mg per kg as a reference drug. Group 4, rotenone-intoxicated rat were protected with pomegranate extract 150 mg per kg per day for 55 days of experiment. The last group, rotenone-intoxicated group, was treated with combination of pomegranate extract and lower dose L-DOPA 10 mg per kg. The exact experimental design was illustrated in the figure. After 55 days of experiment, behavioral tests are conducted for the rats, which is catalepsy test and open field test. Then they are subjected to sacrification for another analysis of histopathological examination of striatum and striatal biochemical analysis of the following parameters: dopamine, brain-derived neurotrophic factor, acetylcholinesterase activity, oxidative stress markers, cyperbutyric acid derivative, superoxide dismutase enzyme activity, inflammatory markers, tumor necrosis alpha factor, and finally apoptotic parameter caspase 3. Results: our results showed that pomegranate extract combination with L-DOPA 10 mg per kg ameliorated the behavioral impairment induced by rotenone in catalepsy test and open field test, prevented rotenone-induced neuronal degeneration in striatum, elevated the striatal level of dopamine, brain-derived neurotrophic factor, and the activity of acetylcholinesterase enzyme, improved biochemical parameters of oxidative stress, inflammatory parameters, and apoptotic parameters. Also, the combination therapy of pomegranate extract and lower dose L-DOPA has shown an almost equivalent effect of that of higher dose. Conclusion: the combined treatment of pomegranate extract with a lower dose L-DOPA have both symptomatic and neuroprotective effect through antioxidant, anti-inflammatory activity, and modulation of striatal brain-derived neurotrophic factor. Pomegranate could be recommended as a disease-modifying therapy when given with lower dose L-DOPA in the early course of the disease. Thank you.